I am Dr. Shipra Goyal, Assistant Professor, Department of Zoology at Kanodia PG Mahila Mahavidyalaya, Jaipur. Today's topic is hematopoietic stem cells. Cells of the body are serviced by two fluids, that is blood and interstitial fluid. Blood is composed of plasma and variety of cells that transport nutrients and waste. Interstitial fluids, that is the cells of the body, they bath in the interstitial fluid. Nutrients and oxygen, they diffuse from blood into interstitial fluids and then into the cells. Waste, that is move in the reverse direction. Then if we see the blood components of our whole body, the 8% is whole blood and 92% is other fluids and tissues. Blood consists of blood plasma, that is 55%, that contains proteins, that is 7%, that is of albumin 54%, globulin 38%, fibrinogen 7%, and others are 1%. Water is 91.5% and other solutes, they are around 1.5%, that is electrolytes, nutrients, gases, regulatory substances and waste products. Now if we talk of formed elements, then they are 45%. Red blood cells, they are around 4.8 to 5.4 millions. White blood cells 5,000 to 10,000 in number, and platelets, they vary 1,50,000 to 4 lakh. White blood cells, they may be neutrophils, that is 60 to 70%, may be lymphocytes 20-25%, monocytes, that is 3 to 5%, and eosinophil 2 to 4%, and basophil, that is 0.5 to 1%. That is the whole component of the blood. There is one practical exercise by which we can measure the hematocrit value, or we can say the packed cell volume. When the blood is taken in a capillary and if we centrifuge it, then we get three layers. The upper layer, it is plasma, that is 55%, and lower layer, that is packed cell volume, that is RBC, that is around 45%, and there is one layer in between two, that is buffy-coat, that is composed of white blood cells and platelets. So the blood elements, they are red blood cells, that is erythrocytes, white blood cells, that is lymphocytes, that may be granular leukocytes. Granular leukocytes, they may be neutrophil, eosinophils, these are eosinophils, these are basophils, right? And there may be agranular leukocytes, that is agranular leukocytes, that means lymphocytes, that is T cell, B cell, natural killer cells, monocytes. Then there are platelets, these are the platelets, they are the spatial cell fragments which is present in the blood. Blood cells, they are formed from pluripotent hemopoietic stem cells. Pluripotent means they are self-dividing and have ability to form the blood cells. Bone marrow may be obtained through these pluripotent stem cells, they come from the bone marrow and that is obtained through aspiration or by biopsy. The sample is then sent to pathology for examination, and the pluripotent stem cells, they form myeloid stem cells and lymphoid stem cells. Most of the blood cell type, why there is a need of a blood cell formation? There is a need of a blood cell formation because they are continuously because they have to continuously replaced, because they die within hours, days or weeks. And thus the process of blood cell formation is known as hematopoiesis or hemopoiesis. It occurs in the embryo, in the yolk sac, liver, spleen, thymus, lymph nodes and red bone marrow, and in adults it occurs in red marrow of flat bones. Now, this is a process how the hemopoiesis or hematopoiesis or or blood cell formation takes place. First of all, there are pluripotent stem cells. These pluripotent stem cells, they are either give rise to myeloid stem cells or lymphoid stem cells. If they are giving rise to myeloid stem cells, then these myeloid stem cells, they uh, form the progenitor cell. This is progenitor cell for erythrocytes, that is colony forming unit erythrocyte. These colony forming unit erythrocytes, they are then forming the precursor cell or blast cell, that is proerythroblast. These proerythroblast cells, they are the formation of reticulocytes, that is nucleus is ejected from the cell, and then there is a formation of RBC, and you all know that RBCs, they are without the nucleus. Then they will be forming colony forming unit for colony forming unit megakaryocytes, that is forming megakaryoblast, and this is forming megakaryocyte, and these megakaryocyte is responsible for forming platelets that we call as thrombocytes. These are forming eosinophilic myoblasts, these myeloid cells, and basophilic myoblasts. So these myoblast cells, they are forming eosinophils and basophils from basophilic myoblast. Then colony forming unit granulocyte macrophages will be forming myoblast, and this will form neutrophil, and monoblast, that will be forming monocyte, that will again form the macrophages. Now these lymphoid stem cells, these lymphoid stem cells, they are giving rise to the blast cells, that is T lymphocyte and B lymphocytes. These T and B lymphocyte cells, they will be forming T lymphocyte and B lymphocyte respectively, right? So they are forming plasma cell. So we are saying that blood cell formation will start with the pluripotent stem cells, and these pluripotent stem cells, these present in the red marrow cells. So 0.1% of red marrow cells, they replenish themselves as they and differentiate into myeloid and lymphoid cell. Myeloid stem cell line, then the myeloid stem line of development continues, that is progenitor cell colony forming units. They will not now divide and specialize to form the specific cell types. That means कि अगर CFU E अगर हम ले रहे हैं, then they will develop into red blood cells. Then next generation is blast cells, right? That is recognizable histological characteristic which develop within several divisions into mature cell types. That is, and then lymphoid cell line development starts. That is pre-B cells or prothrombocytes finish their development into B and T cells. So these lymphocytes in the lymphatic tissue after leaving the red marrow. So we can say that the myeloid stem cells, they are giving rise to RBC, platelets and all the WBC except lymphocytes. Lymphoid stem cells, they give rise to lymphocytes. So myeloid stem cells, they differentiate into progenitor cell or precursor cells. Then these precursor cells, they are forming the precursor cells. They are the blast cells which develop into actual formed elements of the blood. Lymphoid stem cells, they differentiate into pre-B and prothrombocytes which develop into B lymphocyte and T lymphocytes respectively. And this process of forming of all these cells is known as hemopoiesis, and it is actually stimulated by several hemopoietic growth factors, and these growth factors, they stimulate the differentiation and proliferation of various blood cells. These factors regulate differentiation and proliferation, and these factors are erythropoietin, EPO, that is produced by kidneys, and they will increase RBC precursors. Thrombopoietin, that is TPO, this is a hormone secreted by the liver. This will stimulate platelet formation. Then cytokines, they are the local hormones of the bone marrow. They are produced by bone marrow cells. They stimulate proliferation in other marrow cells, and colony stimulating factor and interleukin, they will stimulate WBC production. Erythropoietin does, we can say, increase the number of RBC precursor. Thrombopoietin increases the number of platelet precursors. Cytokines, that will increase the number of WBC precursors. Growth factors, which are available through recombinant DNA technology, and they hold a great potential for use in patients who cannot normally form the blood cells. So, the method of production of RBC is known as erythropoiesis, that is erythrocyte formation or red blood cells formation that will occur in adult red marrow of certain bones. The main stimulus for erythropoiesis is hypoxia. Pro-erythroblast starts to produce hemoglobin. Many steps later, न्यूक्लियस बाहर निकल जाता है, तब रेटिकुलोसाइट्स फॉर्म होता है जो कि ऑरेंज इन कलर होता है, जिसके अंदर कुछ रफ एंडोप्लाज्मिक रेटिकुलम के ट्रेसेस विजिबल होते हैं. अब जब ये रेटिकुलोसाइट्स बोन मैरो से बाहर निकलते हैं ब्लड में, देन इन 1 or 2 days दे इजेक्ट द रिमेनिंग ऑर्गेनल्स और वो क्या बन जाता है, एक मैच्योर आरबीसी फॉर्म हो जाता है. ठीक है. नाउ what is the thing which is controlling the red blood cell production? That is tissue hypoxia. That means when cells, they are not getting enough oxygen, and that is at high altitude where the air has less oxygen. Maybe in the patient which has anemia, that is RBC production, where the RBC production falls below RBC destruction. Circulatory problems. Then when there is a hypoxia, then kidney respond to hypoxia and it will release the erythropoietin. Right here, the kidneys, they are detecting this. Yes, there is a decrease of oxygen, so they will be increase the production of erythropoietin. Now this speed up the development of pro-erythroblast in the red bone marrow cells, right, and they will develop into reticulocytes. These reticulocytes, they enter into the circulating blood and large number of RBCs they are formed, and thus there is a increased oxygen delivery to the tissues, right. Then again there is a return to homeostasis when oxygen delivery to the kidney increase to the normal. So we can say that hypoxia or decrease in the oxygen level, that will causes, or you can say the stimulus that disrupts the homeostasis, that will be the reason of the RBC production. Detection of changes in number of circulating WBC indicate infection, poisoning, leukemia, chemotherapy, parasites, or allergy reactions. So there are two main types of leukemia. Acute leukemia, that means uncontrolled production of immature leukocytes, crowding out of normal red blood cells by the production of immature WBC, prevent production of RBC and platelets. There may be chronic leukemia, that is when the accumulation of mature RBC in bloodstream, because they do not die. They are classified by the type of WBC that is predominant, that is monocytic or monocytic and lymphocytic. Then last is platelets, that is thrombopoietin, that is stimulating the myeloid stem cell and they will be producing platelets. Myeloid stem cells, they will be developed into megakaryocyte colony forming cells that develop into megakaryoblast. Megakaryoblast then transform into megakaryocytes. Then megakaryocytes, that is a fragment. Each fragment then enclosed by a piece of a cell membrane, and we call it as a platelet, that is a thrombocyte. So normal blood contains 1 lakh, मैंने बता दिया आपको 1,50,4 lakh platelets found होते हैं और इनकी जो लाइफ स्पैन होता है that is only 5 to 9 days. Aged and death platelets, they are removed by fixed macrophages in the spleen and the liver. Platelets, they help stop blood loss from damaged vessels by forming a platelet plug. So their granules, they contain chemicals that is promoting the blood clotting. So platelets का होना भी बहुत ज़रूरी है. आप लोगों को पता है CBC क्या होता है? CBC, that is a complete blood count. ज़्यादातर जब आप लोगों ने देखा होगा ब्लड की टेस्टिंग करवाते हैं तो बोलते हैं सीबीसी करवा लीजिए। सीबीसी मतलब कि कंप्लीट आपके जितने भी ब्लड सेल्स हैं, फॉर्मिंग सेल्स हैं, उन सब का काउंट उसके अंदर आ जाता है, जिससे कि आपकी स्क्रीनिंग हो जाती है कि भाई आपकी कोई एनीमिया डिजीज़ तो नहीं है, कोई और वेरियस इन्फेक्शंस तो बॉडी में नहीं है, क्योंकि इसके अंदर आरबीसी, डब्ल्यूबीसी, प्लेटलेट्स क्या काउंट है पर माइक्रोलीटर ऑफ योर होल ब्लड इज गिवन। हिमेटोक्रीट वैल्यू आ जाती है, डिफरेंशियल ब्लड सेल काउंट आ जाता है, हीमोग्लोबिन आ जाता है इसके अंदर। तो ये सारी चीज़ें दे विल कम अंडर द कंप्लीट ब्लड सेल, कंप्लीट ब्लड count. These are the references. Now in a summary, I'll show you a video. Thank you.